see if I'm actually, I made my own screen go away. So let's see. Brave new world of video options. All right, so I'm just gonna go for it, and if they don't see me, they don't see me. All right. So I'd like to welcome everybody to the November edition of Micro Seminar. This is it's been a little bit periodic lately, but we've got one scheduled for today, obviously, and we're always looking for new seminar people to present seminars. And so if you are interested in presenting a seminar, you can be any level, grad student, undergrad, whatever you may want to share your cool research in microbiology. Just please contact me, Dave Baltris, or Cam Thrash, or Lizzie Wilbanks. And so today's micro seminar, I'm very pleased to present Sam Diaz-Munoz. I've actually known Sam for a few years now, coming up through the kind of the experimental evolution microbiology world. He's a great guy. Has always been both an inspiration in terms of the science that he does, which it's really cool, and he's going to show you some of the cool stuff he's doing now, but also in terms of just being a positive impact on both representation in science and from at least my point of view, a really strong force in terms of encouraging both diversity and inclusivity in science in general. That's just awesome to see, and I just want to acknowledge that too. So because I think he's done a great job kind of promoting everything he does with a very positive message, and it's awesome. So Sam started off with his undergrad at the University of Puerto Rico, Mayagüez. After that, he actually went to grad school at UC Berkeley. Actually studying monkey biology, monkey behavior. And so he wasn't a microbiologist in grad school. He studied monkey behavior, and I think that's where he learned about and got interested in sociobiology. And he's taken that to the microbiology world with the stuff he's doing now. After grad school, he did a postdoc at UCSD with Lin Chow and co-advised by Paul Turner. So this is kind of where he learned in his postdoc, where he learned about virology from the Lin Chow, which is an amazing person to learn from. And after that postdoc, he actually went to NYU as kind of an independent fellowship, but kind of working with a couple different labs to, again, add to his repertoire of both knowledge and techniques. And so he was working with David Gresham at NYU on just to learn kind of big data techniques. And then he was able to write a K99 to start working on evolution of flu viruses. And so he's taken that K99 now. In 2017, he started his own lab at UC Davis, where he's been doing great stuff. So he's going to share some of that with you today. And I'm just going to hand it off to Sam now.
Thank you so much, David. I'm really excited. Thank you for that really, really kind introduction. Thank you so much. So I am Sam Diaz-Munoz. I'm an assistant professor in the Department of Microbiology and Molecular Genetics at UC Davis. And I want to acknowledge that UC Davis stands on the land inhabited by the previously inhabited by the Patwin people, part of the Wintun, and I wanted to say buenos días. Mi nombre es Samuel Díaz Muñoz. Soy de Puerto Rico y quería mandar un saludo a mi gente boricua, a mis latinos, pa'lante, and I'm here for whatever you need. So I basically just gave an introduction to who I am, identified myself as Puerto Rican. As David said, I'm really interested in making academia a place for everyone to be here. And so I sent out a message to my community. And so maybe to understand a little bit about why I send out these messages could be illustrated by my first university research experience, which was with this guy, Juan Carlos Martinez Cruzado. And so I contributed really nothing but failed PCRs to this project, but he was very successfully looking at migrations that shaped the Puerto Rican genetic makeup. One of his major findings is that although the Taínos, which are the native people of Puerto Rico, were thought to have disappeared early on, there's actually substantial ancestry reflected in our genome. So representation matters a lot in science for a lot of reasons, but specifically for the research we do and we don't do. It's heavily shaped by us as people and can inform or disinform our research questions, including where we are and where we come from and our ancestry. So some of the most intimate questions that make us people. And so the way I think about this research is that it uses genetics to trace a social interaction. For me, it's all about social interactions. So it traces a social interaction, reproduction among different human groups, and then it uses the record in the genome to see how the population changed to give rise to us as a people. And so thinking about reproductive traits and social traits and how they evolve, for a given organismal trait to evolve by natural selection, there must be some really basic conditions. The person who formally lays these out happens to be Juan Carlos Martinez Cruzado's advisor, Richard Lewontin, now emeritus at Harvard. And he formalized these conditions, and they're now known as the Lewontin conditions. So for an organismal trait to evolve by natural selection, it must be heritable. So it must be passed on from parents to offspring. It must vary in the population, and it must confer differential reproduction, so with regard to that trait. And so today I'm going to frame my talk with this question, asking whether interactions among viruses meet the conditions for evolution by natural selection. And so you'll notice I called the virus an organism. And I did that on purpose in the sense that there are biological entities subject to organic evolution. And so I, along with other people, have written about new definitions of the organism, building on Joan Strassman and Dave Queller's work. And so maybe an easy way to encapsulate it is that I view an organism as a fitness maximizing individual. So I've given you a brief introduction, and here's where we're going to go. I'm going to ask, even before meeting the Lewontin conditions, I'm going to ask, can viruses even have social interactions? Is this even something possible? Do these interactions meet the conditions for natural selection? I'm going to talk a little bit about the development of a nascent field called sociovirology. And I'm going to talk about the importance of variation in science and the scientists who do the science. Okay. Can viruses have social interactions? This is a valid question. It's worth asking because usually this is how we think about viruses. I think most of you will have had the textbook viral life cycle where a virus attaches, takes over the cell, and then they produce a bunch of virus. And so what was really curious to me is that at the beginning of the life cycle, when we started again, it starts with just one virus. Where did all those other viruses go exactly? And what would happen if we started the life cycle with two viruses? And what you can see is that co-infection can change every stage of the viral life cycle, or at least it has the potential to do it. Now that we've gone through this thought exercise, how prevalent is this actually in nature? So for me, this was one of my main questions, particularly because I work with RNA viruses, but I wanted to, with particular RNA viruses in the lab, but I wanted to know how prevalent is this widespread in nature? And I had some sense that this is the age of the microbiome and there's high throughput sequencing. So there should be all this data out there for me to answer this question. And indeed there was. But first I'm gonna do a brief definition of what I mean by co-infection, what I mean by often being in the same host. And so if I have, say, this is a micro seminar, so if I have a flask of E. coli and I put virus A and virus B, is that a co-infection? What's a co-infection in some sense? It's a co-infection of that multicellular microbial culture, which I'll call culture co-infection. And then there's a type of single cell co-infection, which is what I showed in the life cycle at the beginning, which is a single cell with more than one virus in there simultaneously. And then there is something that most call cross-infectivity or phage host range, which I have relabeled potential co-infection. That is, different viruses can infect the same host, but they don't necessarily do so at the same time, or we don't know if they do. So going back to that question, it turns out if you look at virus-host interaction data sets in the cellular microbial world, what you'll see is that half of all microbial hosts are infected by multiple viruses. And so I'll show you this table, which is actually table 1 in the introduction of my paper in virus evolution. And the reason it's there is because this data was out there, was known, and so I'm just sort of collating it all together so we can answer this question. And what you can see in the third row there is that of all the microbial hosts, microbial cellular hosts that were, so bacteria and archaea, mostly bacteria, that were infected by viruses, about half of those are infected by multiple viruses. And when you look at the average of that, it's not just like one more, like 1.2 or 1.3. Even at the single cell co-infection level, we're talking 2 or more on average, right? And so another thing that I want to highlight is that this data set it's basically a collection of 3 different data sets that encompass sequencing and culture-based methods of detection, and thousands of viruses, thousands of hosts in different environments collected at different times, right? And so I think it's pretty remarkable that we have that consistent pattern in co-infection, and there have been subsequent studies that have shown sort of similar patterns there. So what I actually wanted to do, knowing that now that co-infection is something that is prevalent, is what explains the different levels of co-infection that we see among different hosts. Is the host important? So does it matter if it's an E. coli or Pseudomonas aeruginosa? Does it matter where the host lives? Do the viruses themselves play a role? And so I wanted to just highlight that first, it turns out the type of ecosystem where a bug lives is pretty important and it's going to shape co-infection. So I'm going to walk you through this. On the left, you have a data set that's about potential co-infection, so cross-infectivity. And on the right, you have a data set that's sequence-based, which is about culture co-infection. And so what I want you to see, what I did here is I used the JGI Gold database to put these on the same sort of ecological ecosystem classification. And so what you'll see first is that there's the most co-infection in hosts that are associated to bigger microbial hosts. So this is your like gut microbiomes and your plant microbiomes. That is the highest level of co-infection. So I just normalized that to 100%. And after that, the second level, the second ecosystem with the most co-infection are engineered habitats. So this is your yogurts, your bioreactors, all that. And the lowest is the environmental ecosystem. So this is what we actually think of as sort of natural ecosystems, ocean, soil, all that, right? And so what I want you to notice is that not only is the rank order of these different ecosystems in terms of co-infection the same, but actually those percentages are pretty close to each other, despite these coming from different data sets. So the implication of this is that the ecological factors that affect whether viruses can infect the same host are the same ones that affect whether the host is actually co-infected, at least at the culture level. So far, I've been just basically counting viruses and hosts. And so what I wanted to do was look at, is there evidence that the viruses themselves interact, right? And so what I found was in 2 of the data sets, the viruses had been identified by sort of their infection lifestyle. So some viruses integrate and other ones have a lytic or sort of latent chronic infection cycle, right? And so in these data sets, primarily led by Simon Roux, who's now at JGI, those were identified. And what I found was that prophage sequences can limit co-infection by active viruses. So what you have here on the y-axis is the number of active viruses that are not integrated into the genome. And on the x-axis, you have the number of prophage sequences that we find that are integrated into the genome. And what you see is as you increase the number of prophages, the number of active viral infections actually goes down. And this happens both at a culture co-infection level and at a single co-infection level. So now, is there evidence that the identity of the virus matters when they interact? And so something that I did was look at one way, so there's massive amounts of what they call viral dark matter, which is, we know there's all this viral sequence, but we don't know what they are. So one thing we do in the field is we arrange them in clusters. Those are normally pretty equivalent to genera. So we'll call them viral genera for the remainder of the talk. And what you have here is a histogram. And what it's doing is taking all the co-infections that I saw, all the co-infected bacteria and archaea that I saw in the data set. And I'm asking how many viral genera are represented in each one of those co-infections. And so what you can quickly see is that the same viral genus co-infections are rare in all co-infections, right? So they tend to be, there tend to be more that have two, and then it sort of goes down, right? Another way to see that is to flip it to the viral perspective. And so what you have arrayed here on your y-axis is all the different viral genera in this data set. And on the x-axis, you have the mean number of viruses that are co-infecting. So what you can see very quickly is that the dots, which are the means, are really in between this box that's in between 1 and 2, save for a couple of exceptions. Not only is the mean, but even you can see even the standard deviations are almost within that box. And what this suggests is that viruses rarely have a co-infection partner from the same viral genus. Now, I'll add a quick caveat that this data set, because it's based on sequences, it's based on consensus sequence data, it excludes really closely related viruses, which are strains, right? And so I'll actually talk about that in the next part of the talk, where I use my study systems in the lab, in the wet lab. Okay, so let's recap. Can viruses interact? Well, viruses are often in the same host. This should be well known, and I'm happy that the paper is being cited to this effect. And other things that we have found are that the extent of this co-infection varies and can be shaped by the actual bug being infected, by the ecosystem of the host, and the viruses themselves and their interactions can set how many other viruses co-infect. And there's evidence that these interactions can be really specific. Okay, so moving along, as I mentioned, I'm gonna move over to the wet lab part of my research and ask, do viral interactions lead to meet the conditions for natural selection? Okay, so a little bit of background here on an interaction that has been the focus of my postdoc work going into my lab, and that is genetic exchange. So here, this should look a little bit familiar from the cartoon life cycle. I have what's called a segmented virus. So its genome is arrayed into 3 different pieces in the case of the phages that I study. So you can think of them roughly as chromosomes. So upon co-infection, say co-infection of 2 strains, now we have 2 distinct lineages, 6 segments in the cell, in the case of these phages. And when those segments are swapped, this is called reassortment. And we use that word to distinguish it from recombination. There's no homologous, or I should say, there is very little evidence of homologous recombination. It's basically just the wholesale segment swap. So this exchange is clearly an interaction. And from a social perspective, the way an evolutionary biologist likes to look at this is to look at the cell as a shared resource, right? And when there's a shared resource, this leads to the conditions to foster conflicts of interest. So this would suggest then that genetic exchange would be sort of tuned to get sort of an evolutionary advantage. However, what I found out in my postdoc looking at the literature is that in virology, that wasn't necessarily the view. So the view was that the genetic exchange would be strongly constrained by the molecular mechanisms of packaging. So this sort of lab rat virus that I study, Phi6, has this really elegant packaging system to make sure that it has 3 copies of its entire genome, of all its segments in the capsid, and it does it sequentially, and then the capsid maturation follows this sort of inflating beach ball model, right? And so the thought was that for cystoviruses and viruses in general, that genetic exchange would vary according to the genomic architecture of the virus in question, and not as a trait that would be optimized by natural selection for its own selective value. That is, reassortment is a fixed property of the genome. And so to me, that didn't sound quite right because ecology and because evolution. And so what I did for my postdoc work is I isolated different relatives of this virus Phi6 from plants out in the wild. And what I found was that depending on where these viruses were originally collected, there were differences in the level of reassortment, rather in the level of genetic exchange or in the proportion of hybrids, which is what I actually measured. So then started figuring out that reassortment is actually pretty dynamic and complex, and that this interaction does meet at least one of the conditions for natural selection, which is variation. Now, you have sort of a similar issue in the influenza field. And I had the opportunity to move into eukaryotic virology with my postdocs at NYU, where I work with David Gresham to learn high throughput sequencing, and then with Elodie Gedin and Binzu, who's now at CDC, to learn about flu. And so what I learned from them is that if you ask experimentalists, influenza experimentalists, can you take strain A and strain B and get reassortments in the lab? They say, no, this is extremely difficult. They are rare. And so that whole literature has evolved around the idea, or has been framed around the idea of constraints. Well, perhaps these segments are co-adapted, or perhaps there are differences in the viral fitness. And so that's sort of the way it's gone along, right? And so it seems sort of weird that there's a contrast with what we know from epidemiology. So we know influenza is one of the biggest, it is pretty much bar none the biggest killer in terms of pathogens. And quick tidbit if you didn't know, because I was researching around the anniversary of the 1918 pandemic, and I knew that a bunch of the world population, 4%, had died, but I didn't know that half of those deaths happened in a 10-week period in the fall of 1918. So that's about a quarter here at UC Davis. So that's pretty stark reminder of the power of this pathogen. But those pandemics, which happen recurringly, are sparked usually by reassortment between avian and human strains of influenza, right? And our seasonal epidemics are milder, if you will, but they still kill a bunch of people. And they become worse when you have reassortment causing vaccine mismatch, as happened in the 2017-2018 pandemic, right? So is it possible that these social interactions that we see in a single cell in the lab are rare, and then they have global consequences when we don't really see them in the lab? So it led to the question, is influenza genetic exchange rare? And so there was this really elegant, thorough study out of the lab of Anis Lowen, who's at Emory University, that said, okay, there's supposed to be all these constraints to reassortment, so why don't we make virtually identical strains? So they introduced synonymous substitutions into a particular influenza strain, verified that the replication kinetics were good, and then said, let's do some unbiased conditions and reassortment and put these two strains together and see what do the offspring look like. And lo and behold, there is a ton of genetic exchange now, right? And so I'm gonna walk you through this plot because I'm gonna show you a couple of plots that are like this. And what you have is in each of the columns, you have one of the 8 influenza segments, genome segments. And each of the rows represents one viral progeny. These were isolated plaques, so this is analogous to a colony. right? So you take strain A and strain B together, one cycle of replication, and then you genotype the progeny. You can see just visually, this is basically a shotgun blast of red and green, which are the parental strains. And indeed, the level of genetic exchange here is actually higher than we expect from random. So this shows that under completely unbiased conditions, you can actually get really free genetic exchange between influenza viruses. However, for me, I was interested in looking at different strains because I'm interested in diversity, but also because some of the interesting things about reassortment is when two different strains come together. So what does reassortment look under unbiased conditions with different strains? And so what we did was do pairwise crosses, so co-infections between a diverse panel of seasonal influenza strains. And this was work that would have not been possible without this group of people. Ileago is a grad student in my lab. I was as our project scientist, and Kishana Taylor is a talented postdoc. And they took these 6 strains, and to look at the generation of reassortants from this diverse panel of strains, we had to get a little bit creative with our high throughput sequencing approach. And so I won't go into too much detail about the approach, but basically what it does is instead of providing high coverage of relatively few individuals, this approach can actually provide low coverage of thousands upon thousands of individuals while preserving genetic linkage. So if that's something you're interested in, we can definitely go into that or chat a little bit later. But what I'm showing you is unpublished data from about 1900 clonal isolates from 20 infections, 8-segment genotypes. And so our data set right now with the manuscript we're preparing is probably about double this, this data set. So this is the plot that I showed you from before, sort of the state of the art right now. And so what we can do is generate a similar plot, but use a diversity of strains. And what you can see right away is that there is genetic exchange going on between different strains, despite their different origins, their different antigenic subtypes, and all this stuff. And what I'm going to do is I'm going to take each one of those plots and basically summarize them in a bar plot as the percentage of reassortants that were generated. So strain A and strain B, how many of those progeny were of mixed origin? And so that's what this plot is showing. And I want you to notice, this might be obvious, but I just wanted to emphasize that the way these experiments were done, these are the same host cells, same conditions, and within one cycle of replication, you have a complete rearrangement of the genome. So that's a different population. And as you can see from the variation, this is due to the identity of these strains. So each one of those bars is the result of the crossing or the co-infection of strain A times strain B, and on down the line. Okay, so we can have strain A and strain B generate sort of a high level of reassortants, as you can see between this blue and green strain combination, and you can have a low percentage of reassortants. But this is differential fitness sort of in relative terms, and what is a percentage of progeny produced, but it's not the whole picture, right? So the best way I usually think to illustrate that is thinking about we're approaching lunchtime, and if you have a friend that offers you, 5% of their pizza versus a friend that offers you 10% of their pizza, you're probably willing to go with a more generous friend that's giving you 10% of their pizza. But what you don't know is what is the size of their pie, right? You might be disappointed. And so this is sort of what happens with influenza. So not only the rates of reassortment affected, but the actual replication rate is also affected, such that if you had a strain pairing that actually generated a lot of strains on a relative percentage basis, that it actually replicates less, there's less efficient replication, so then your total reassortant progeny is actually lower than a strain combination that yielded few reassortant progeny on a relative basis, but generated more progeny in total, right? So it's really important to keep both of those things in mind to figure out how are the interactions between strains going to affect the populations going forward. So I posed this question earlier, and now I think what we're moving towards is the idea that it's actually these viral interactions in single cells are not rare, but rather they happen pretty frequently. And so why we don't see them necessarily is because subsequent selective factors such as post-adaptation or immune evasion sort of erase that variation that happens. And so the important thing is to distinguish between the within cell interactions to the between cell interactions, because if we're talking about reassortment and measuring reassortment, we want to only look at those within cell interactions. And indeed, this is something that was mentioned early on in the first studies of influenza virus reassortment, that there had to be a tight control of the cycle of replication. And so this is something that we think is explaining our results. So, and I'm just going to show you one of these plots, and I'm going to zoom in to these three segments that show the effect a little bit better. So one way to control replication to be only to one cycle is to limit the time to 12 hours, right? But another thing that is done usually is you can withhold trypsin, which is a protease that the virus needs to cleave from the cell receptor and bud and go and infect other cells, right? And so those are the conditions that, for example, Anise Lowen used in her experiments and that we're using in ours. And what we tried to test was, well, what happens if we relax those conditions? So if we add trypsin, but still keep it to that 12-hour time point, what you can see is you start detecting way fewer reassortants, and the blue just starts dominating this, the progeny. And if you extend the timeline even more, then you'll see that your reassortants indeed become rare. So I think it's a combination of tightly controlling the conditions and the resolution of our approach that allowed us to really quantify reassortants among different types of strains. And so the news sort of for the influenza field in general is sort of terrifying and hopeful. It's terrifying because it means if two viruses are in co-infection, pretty much we'll get a pretty good representation of pretty much every card they can deal us. But the hopeful, which is terrifying, but the hopeful part is they really need a specific selective environment to actually go global. Okay, so future directions in this, we just actually today received some viruses that we will use to investigate avian influenza virus reassortment, and that's going to be led by Dr. Kishana Taylor. And I'm really excited about using this reassortment data set to look at the molecular mechanisms of influenza viral genome packaging and how all these segments get into a capsid to form an effective virus. And I really am excited about leveraging the diversity of the strains that we have to answer this question. And on the phage side, thinking about whether phages interactions between viruses meet the conditions for natural selection, this is some work being done by Sari Mantinen, who's a talented postdoc in my lab who is now a fellow of the Finnish Academy. And she is going to look at whether the social environment affects reproduction. So can we isolate the social environment as a variable? And this is in collaboration with Dan and Weissman, where we're doing some modeling to look into this. So coming back to our initial questions, our initial conditions, we have some evidence that there is some heritability in the sense that there are characteristic reassortment patterns depending on the identity of the strains. We have some more work to do there. there's definitely variation in the interactions in genetic exchange. So we see that very, very clearly right off the bat. In a single cycle of replication, there are differences. And there's definitely differential reproduction. So those interactions are going to lead to a relative level of genes being transmitted, so the actual allele frequency, and to the actual number of progeny into the viral replication. Okay. So as you saw, we use sequencing and bioinformatics to answer our questions about the social lives of viruses. And we have to tweak stuff sometimes, and that leads to opportunities for developing new things that may be useful to other people, and to collaborate with other people to help them tackle their problems. So I wanted to do a little bit of what I'll call a techniques interlude, where we have two projects where we've been developing some techniques for viral detection. So as microbiologists, sequencing is an amazing tool for us, right? We can do culture independent stuff. We can chart a lot of different places. And now, a bias that we need to think about in metagenomic studies that's obviously different from culture is the type of nucleic acid that we're sequencing, right? So, for example, if we don't sequence RNA, we might miss half of the viruses in the ocean. And if we get into more specific types of nucleic acids, like double-stranded RNA viruses, which we happen to be very interested in, we might miss them if we're doing microbiomes and metagenomes. And outside of the microbiology world, or sort of peripheral to the microbiology world, but still related, double-stranded RNA is important in host defense against pathogens in eukaryotes and in plants. So this is something we wanna know about, and people were having a lot of difficulty. So the Phi6, which is the strain that we study here in the lab, one of the main reference strains, work of Brett Durakoff and Laura Hooper at UT Southwestern, they were trying to assemble a mock microbiome and to look at all the different viruses that were there. But even though they deliberately knew that they had Phi6 in there, they could not retrieve a single read from it, right? And so here's another recent study showing that basically they're getting 30 to 70% of the reads mapping to the host and not to the virus. So this is obviously a barrier, and I'm not putting these examples here to downplay anyone or downplay anyone's efforts. For us, it was the same thing. We were just as miserable. So this is a plot that shows the percentage of reads that map to a reference genome when we sequence the reference genome, and you can see that if we just use untreated lysates, we got basically nothing. And we tried to concentrate, and that didn't help. And so then we're like, okay, let's use a nuclease treatment to eliminate all this rRNA that's in there, and that made things a little bit better. So then we're like, okay, maybe we can concentrate a nuclease, treat it, and maybe that'll be better. But then that ended up being worse because we concentrated the contaminating rRNA as well. So, we're really trying to figure out how to do this. And it was a postdoc in my lab, Alex Wilcox, that really cracked this problem. And I wanted to mention that he is looking for a postdoc. He's a good person to have along. And he was investigating and reading. And so it is well known that DMSO is a denaturing agent for double-stranded RNA. It's been used for RT-PCR, and it had been used for Sanger sequencing. But as far as we could tell, no one had used it for next-gen sequencing. So Alex said, let's just add DMSO. And lo and behold, even if you have non-detectable amounts of RNA by qubit or cDNA by qubit, you get an amazing increase in reads. So this is like a really cheap chemical, and we can add it, and basically it's more important than concentrating your viral sample. So Alex did some extra work to look, and he actually sequenced influenza using the same protocol, and he saw that it doesn't affect the recovery and coverage of a single-stranded RNA viral genome. These are the 8 segments of influenza and the coverage that you get according to the different treatments. And so that doesn't change. And it also doesn't negatively impact the sequencing accuracy in flu or in the double-stranded RNA viruses. So for all you microbiologists, this is a micro seminar. If you're doing a metagenome and you want to look at the RNA, add the MSO. It won't affect your ssRNA. As far as we know, it won't affect your transcripts or anything like that. And you'll probably get a huge increase in the double-stranded RNA viruses, which are very uncharacterized. And if you have friends that are more on the immuno side, please tell them about this method, because I think that this is a method that would be really useful for them. And I fear that it'll stay sort of in our microbiology silo. Okay, so the next thing that I looked at that we had an opportunity to contribute to was detecting avian influenza virus in water. Okay, so in the interest of time, but perhaps at the risk of being crass, birds are the natural reservoir of influenza virus, especially these ducks and these waterfowl. Influenza is a digestive disease, birds shit in the water, and then the viruses can stay infected in there. So if you're interested in keeping poultry safe, say, from these high pathogenic avian influenza outbreak and not trying to kill our food supply, and if you want to monitor detection of potential pandemic strains, as I mentioned, they happen between avian and human influenza reassortment, then you might want to know what's in the water. Now, the standard method for that now is to take the viral genome segment and do a qPCR of 100 base pair amplicon in there, right? So when Maurice Pitesky and Madeline McEwen, who's a master's student in his lab from veterinary medicine here at UC Davis, came to me asking, what is another way that we can use for detect? And I said, well, why don't you take PCR up the entire segment, and then we'll just try to throw it on a minion and see what sequence we get. And my reasoning was that the ends of the viral genome, which is this viral segment signal that I'm depicting up there in gray, those are really conserved among all influenza A viruses. And so what we found briefly is that we get a huge increase in the detection of influenza from water samples, of avian influenza specifically. And so in contrast to 100 base pair amplicon, we actually have amplicons that are basically as large as the segment in many cases, and we can match them with high identity to a known avian influenza database, right? And so what that enables us to do then is to try to make predictions of potential host species and to ascertain the antigenic subtype, sort of in the one, using just one test, basically. So some of the take home from this is what we think is, because we get these amplicons that are so long, we think that we're getting nucleic acid that comes from the capsid, because we don't think that 2.3 kilobase pair pieces of RNA are just floating in solution. And so as always, when you're sequencing something and blasting it, you have to be careful with inferences about a potential host or a subtype, but it's a further tool that we have. And what we found is that primer conservation is really crucial, and I think it underlies the difference that we get in the qPCR method that's standard now and the sequencing, the whole segment approach that we use. And to that point, what we did is we went and tested whether that standard qPCR method would work with the known avian influenza virus genomes in the database. And it turns out that a lot of times it's predicted to fail in the vast majority of cases. So that's something that I'll update you on the Twitters, and we should have something to present formally about that soon. Okay, so I wanted to talk about a little bit about sociovirology, which if you're on Twitter, you saw that that's my handle, and to answer sort of a couple of frequent questions that I get. One is, is this new? We know that this happens, right? And that is absolutely true. And I love putting up this paper by Renato Lulbeco that has such a delicious sociovirological title, which is mutual exclusion among related phages, right? And so this is an area that's still under research, And so I'm aware that this happened before. And actually, the origin of the discovery of interactions between viruses actually extends even further back. So if we look at the Nobel Prize from 1969, Hershey, Delbruck, and Luria had showed that phages could co-infect and exchange genes, right? But in an interview that Ellie Dolgan made for Nature to Dale Kaiser, who was one of Delbruck's proteges, the way he described it was, even though they were using terms like cooperation, they were thinking about the phages as an experimental tool, right? And so not thinking about what they said about virus ecology. And so here's the quote: "It was a way of making a genetic cross between two virus strains." The moment in the literature that I peg as sort of the birth of thinking about social interactions between viruses is a paper by Paul Turner and Lin Chao in 1999 that's called "Prisoner's Dilemma in an RNA Virus." And so these were my postdoc advisors, and what they did was they used data from viral co-infections to parameterize a game theory model that's often used to explain the evolution of social behavior in everything from humans to cancer cells. So why do we go from 1940 to then 1999 to now, where there's a little bit of an explosion of viral social research? And so I think it has to do with being focused on viruses. We know much more about viruses. We're interested in viruses for their own sake. We know more about the molecular mechanisms, and we have a critical mass of researchers. And so what I want to do here is go over some of the findings that have been coming in recent years, really in the last 5 years, basically. And these are not citations. These are more the groups that have led some of these things. And I'm going to leave out a bunch of people. I really want to emphasize that there's a lot of people doing this work. And so we might have not, they might not all be saying sociovirology, but they are doing it, and we're reaching this critical mass. So we've found that viral co-infection is frequent. I talked to you about that at the beginning. We have found that viruses, as a community, the viruses infect together, and this has been work by Nihal Alton-Bonet at NIH and Rafael San Juan at Valencia, and later Julie Pfeiffer's group at UT Southwestern. And viruses literally stick together and infect as units. We found that viruses have their own language, they communicate, they have a quorum sensing-like system, and this is work by Rotem Sorek out of Israel. And not only that, they can actually eavesdrop on the quorum sensing signals of their bacterial hosts, as shown beautifully by Bonnie Basler. Also, viruses can interact. Really nice work from Catherine Jouet and Jesse Bloom's lab. Edza Westra and Joe Bondi-Denemy have done some amazing work looking at CRISPR. And we also know that co-infection shows reproduction, as I've shown you work from our group and from Annie Sloan's lab. And so where we wanted to step in was really to see that there was a new field where social evolution theory could be applied and was being applied, and sort of provide a framework for that. And so that's our sociovirology paper, and it's mainly couched in evolutionary terms, but really we think sociovirology is larger than just the evolution, but it provides a convenient framework to understand viruses. And so what we want to do is highlight what is going on to build community. And so I co-organized a symposium with Asher Leeks from Oxford University, who's a student of Stu West, and this symposium was called The Social Lives of Viruses. And we brought in a really diverse set of speakers. And I felt like the goal was accomplished when I had one of the scientists say, I really didn't think of myself as doing sociovirology, but I guess I do. And so that's really the point of what we're trying to accomplish, to build a field and sustain it, and make sure that we're converging on the generalities and pushing the field forward so we can discover more about the social lives of viruses. All right, So now I want to talk about the importance of variation in science. And I think that means by extension and necessarily variation in scientists. Okay, so we have an issue with, we all know that diversity is a central concept in biology, right? Without diversity, there's no evolution, leads to extinction. We all know that. But where does that diversity come from, right? And here's a little drawing from the Learning Evolution website at UC Berkeley. Sorry, I forgot the attribution there. But basically, you have these bugs, right? And you grow them on different foods, and eventually they become adapted to that, right? And so they diverge and they garner new abilities. And then with gene flow, with genetic exchange, you can commingle those abilities and lead to new combinations, right? But that local adaptation is important because it's the source of that diversity. And if we think about academia and our local environment, we need to think about what our local environment is enriching for. What, to draw an analogy to microbiology, what bugs is it set up to support? And so if we think about selective media, we can really sort of grasp that analogy, right? And so what we know is that our institutions, our physical institutions especially, were designed for a particular group of people. And so this is in part led to our diversity problem right now in academia. So how do we create inclusion when institutions isolate? One organization that I've been involved with is Ciencia Puerto Rico, which is a social network of everyone interested in science in Puerto Rico. And it really brings together the social, the Puerto Rican scientific community through social networking. And so there's over 10,000 members right now. The organization passed its 10th anniversary, and there have been a number of initiatives that have arisen organically from the community, and the organization has received a lot of recognition from everyone from the White House to the Union of Concerned Scientists. We have a fantastic board of directors that is invested in our mission, and we have wonderful programs to help grad students, minority grad students in school. So there's all this stuff, and we write about this approach in a paper in PLOS Biology 2013. I encourage you to check that out. But the point is that we're creating sort of that network that we don't have physically, because most of the time, minorities by definition are the only ones there, right? And so this should not be so foreign to us. Making spaces for a particular culture is not weird. That's exactly what we're doing here at Micro Seminar, right? I am blessed to be an institution that has a ton of microbiologists. But some of you might be in an institution where you're the only microbiologist, right? And so you need that community. So Micro Seminar is part of that community. That's why we go to conferences, for example. And there's a parallel to our own communities of origin that were not thought of when they built universities. And the same way that Micro Seminar is a space for microbial culture, minorities need spaces to be themselves and to really be able to bring the diversity to the table that's going to enrich the research. And so how does this affect research? So this actually directly affects research, and in particular microbiological research. But I'll start with an example from human genome research and go back to Juan Martinez Cruzado. And what he has been involved in the 1,000 Genomes Project, and himself, along with Carlos Bustamante from Stanford and others, had to actually lobby hard for the inclusion of Caribbean people in the 1,000 Genomes Project. And so the ancestry map that I showed you before of my ancestry would have not been possible because of that. And all our inferences are dependent on that. And in microbiology, we're actually really close to that. And we think we need to think about specifically about the microbiome. Who are we studying? And we're in an early enough stage that we are encoding right now what is normal. So the populations that we study are really important to establishing this normalcy. And this is a point really driven home to me by Larry Forney in several of his presentations, where he's done some amazing work examining minority populations and what their microbiomes look like, right? So this is really, really important. And likewise, we have to think about which environments are we studying, and what questions and techniques are we prioritizing. So I wanted to make this a little bit more concrete by presenting another project that's in an early stage in my lab. I presented at the Gordon Conference for Viruses and Cells earlier this year, and I just wanted to make it as a concrete example of what I'm talking about. Okay, so moving into the influenza field, it was amazing to see that there's all this genome sequence, right? And in particular from humans, there's a bunch of data collected to understand the virus and to design vaccines, right? But and that is really, really useful information. So there's an estimated 22% reduction in mortality due to this influenza surveillance, right? But what you'll see, and particularly I wanted you to look at this bar plot on the left, the one with the filled blue bars. There's incredible bias in where these sequences are collected. And this is on a log scale. And you'll see that at the end of that first bar plot, those two little bars at the end that you almost can't see are Africa and Antarctica. Okay, so there, and again, the Antarctica sequence, this is human viral sequences. This is not penguins. These are human sequences, and it's not like there is not influenza in Africa. So this has a consequence. And in a recent study, this has been quantified. The gaps in influenza surveillance where we don't have genome information, that results in higher mortality. So people are dying because of this. They are hit harder by influenza. And so this really contrasted to the world that I've been exposed to in science, where it's all about declining sequencing costs, right? And access to bioinformatic tools. So can we use tools like the Minion sequencer that I have right here in the palm of my hand? And can I use bioinformatic know-how that is accessible to me here at UC Davis to improve genome sequencing and surveillance in low to middle income countries? And so I want to emphasize that this is not just a question of helping people, but about biasing our understanding of the fundamental ecology and evolution of biology of influenza virus, sort of at our own peril. Those viruses are there, and we're basically just closing our eyes and ignoring them. So from both perspectives, I think it's important to look at both those things. Okay, so just a quick recap. I hope to have convinced you that viruses can have social interactions, that they are indeed in a host many, many times, and that those co-infection patterns are shaped by the host, by the ecology, and by the viruses themselves. And I think we're gaining ground on outlining the conditions for natural selection in terms of at least in terms of genetic exchange. I hope you saw that we're trying to build a new field that will really lead to new insight in terms of the social lives of viruses, but to virology in general. And hopefully you have seen throughout the whole talk, I've been talking about diversity, I've been talking about variation, I've been talking about interactions, and that is important for the science and for the scientists as well. And so I wanted to end by thanking my lab group. They are an amazing group of researchers and an amazing group of people. And I feel really, really blessed that they have entrusted this part of their career to me as a new BI, and I don't take that commitment lightly, and I hope they're advancing, and they're certainly making some amazing discoveries. I wanted to acknowledge them and all my collaborators and funding sources, etc. And so I would love to take your questions.
Cool, that was great, man. Thank you. So I do have one question right off the bat. So Alex Wilcox, you plugged him as looking for a postdoc. Can you give the details as to maybe Twitter handle or how to get in contact with Alex?
Oh, fantastic. Yes, you can contact him at Phageologist. So a studier of phage. And that he is.
All right, I will add that to the tweet I sent out with that. So I didn't see any other questions, and I'll keep monitoring. But I've got a few that just popped up in my own head because I'm a bacteriologist, and so some of this stuff is a little bit foreign to me. So it's cool to kind of get another perspective on it. All right, so the one thing I always hear is that we're worried about these world flu pandemics where H7N2 or other things will recombine and actually turn into the 1918 flu again. That we'll get these other pandemics that wipe out a bunch of people. Your data suggests that recombination is happening a lot, and then it's just selection after the fact that's kind of limiting it. So are these pandemics not happening that frequently because of our own bias and how we're just not seeing it, or is it the recombinations are happening and there's selection against it? Or is it just that the viruses aren't present in the same cells in that case, so we don't get those crazy recombinants that would lead to the global pandemics that we're worried about?
Yeah, so I think there's a little bit of truth to the last two things that you said. I think the frequency of them has been pretty consistent throughout. So if you look, and so the most recent one was 2009. So we do get those pandemic events. Obviously, none of them has been as severe as the 1918 one. And with 1918, there were a bunch of other sort of aggravating factors in addition to influenza. So, that's not my field predicting when that happens. But in terms of the genetic exchange, I think that it's absolutely right that there is probably way more genetic exchange than we think going on. And then it's the selective environment that's not right. If we think about a pandemic, several things have to happen, right? So avian influenza viruses are really crappy at infecting mammalian cells in the first place, right? But they are pretty pathogenic. And then the human strains are sort of the opposite of that, where they can spread mammal to mammal very quickly, but they're not particularly pathogenic. So you have to have both those strains come together and produce a reassortant that then is capable of replicating well in a mammalian host and also be pathogenic. And usually that involves some intermediate host, which is usually a pig, which is why it was called swine flu. And they call them mixing vessels for their permissiveness for this. And then it has to jump. So there's several jumps, right, that this virus has to do, which involves a lot of adaptation in each of those subsequent steps. So I think that's one factor. I think something that we haven't thought about as much, which is what you mentioned at the end, is, well, maybe they never get the opportunity to co-infect, which is a great point. And so what I'm hoping that we could do is, I mentioned that we have some work that we're planning on doing on the molecular mechanisms of packaging. So I'm hoping some of that will be able to arrive to a sequence level predictability of reassortments. So be able to just look at genome sequences of strain A and strain B and say, this is sort of how likely we think it is that these strains will reassort based on the packaging regions. And so if we have that, then we can actually sort of monitor that. And it wouldn't be, it'd be a challenge, but I think it would really complement really nicely the real-time surveillance that we have right now. Because right now you can go to sites like nextstrain.org. So this is a site led by Trevor Bedford, among others, up at Fred Hutch and other places. And I encourage everyone to go there. It's really amazing. So they have Next Flu, but now it's brought into Next Strain. So for a lot of viruses, you can see sort of in real time, how are these viruses changing? And there's trees that are dynamic, and you see where the strains are isolated from and all that. So I think if we can add that extra layer, I think that would be really, really valuable. So that's a great point.
Next question I have. So you mentioned the PCR of the whole segment and sequencing on a MinION actually does better at surveillance. So why do you think, is that just a mapping thing? Are you just getting more information and you're able to map, or is there something inherently better about sequencing the whole segment?
I think it's a little bit, it's almost like a little bit of an unfair comparison, but basically it's about primer conservation and conservation of sequence. So this qPCR was designed to target the matrix protein of the virus, which tends to be the most slowly evolving one. But if you ever looked at a viral alignment, you know there's variation everywhere, right? And so over time, that area is under selection, so it's going to drift. Now, one area of the genome that's particularly well conserved are the packaging regions at the very end. So they have 12 nucleotides and 13 nucleotides at the end of each segment. And these are conserved among all influenza A viruses. And so that's actually what we use in the lab to amplify the whole genomes. And this was a technique developed by others, by Benzu, who trained me at NYU. And so it's really, really powerful. You can just pick up those 12 and 13 nucleotides, and you have your whole genome amplicon. So I figured, hey, if we have that, and I had been doing some barcoding for the genotyping work, let's just barcode them and let's just throw them on a minion, and that should be really cost effective. And so it did work out pretty well. And so the sequence part is sort of extra, right? You know, I think with the per base pair accuracy, which people obsess over, I think a little bit too much, with the minion, you still have to be careful if you're going to infer like a host species, or if you're going to infer a particular subtype or stuff like that. But I think that's a really good technique to use. And we basically, to look at whether that was the case, we actually run basically like simulated qPCRs on existing data, and what we find is that at least with the primers that are published, it's really hard to get it to work with the flu sequences we know exist, right? So it suggests that there might be a different approach to go there.
Cool. All right, I'm going to take one more cruise around and see if there's any questions anywhere. Doesn't look like it. So just thank you again, man. This is awesome. I will try to package this and put it up, and I'll get it up on the website and tag you in the links when I tweet that out as well. So it was great. I like the topics kind of you went over. You hit a lot there, and it was awesome. So thanks much.
Thanks so much. Thanks, David.